Hello and welcome back to NEET Prep. So today we'll be doing molecular basis of inheritance. Without any further ado, let's get started. In the previous chapter, you have learned the inheritance pattern and the genetic basis of such patterns. At the time of Mendel, the nature of those factors, or now that we call genes, regulating the pattern of inheritance was not clear. Over the next 100 years, the nature of putative genetic material was investigated, culminating in the realization that DNA or deoxyribonucleic acid is the genetic material. So over the next 100 years, so earlier the nature of the factors or genes regulating the pattern of inheritance was not clear. That is, what was getting inherited, how it was getting inherited over generations. So over the next 100 years, the nature of those putative genetic material was investigated, culminating in the realization that DNA or deoxyribonucleic acid is the genetic material, at least for majority of organisms. In class 11, you have learned that nucleic acids are polymers of nucleotides. Deoxyribonucleic acid (DNA) and ribonucleic acid (RNA) are the two types of nucleic acid found in living systems. So, after over the next 100 years after Mendel, after he described the factors and all, it was the genetic material was investigated, and the culminating in the realization that DNA is the genetic material for most of the organisms. Why most of the organisms? Because in viruses, the genetic material is RNA in many of the viruses. So, except for viruses, which is an exception, in all of the other, and in fact viruses are living or non-living, even that is not clear. So in most of the or majority of organisms, DNA is the genetic material. Then deoxyribonucleic acid, DNA and ribonucleic acid Deoxyribonucleic acid (DNA) and ribonucleic acid (RNA) are the two types of nucleic acids which are found in the living systems. Now, DNA acts as the genetic material in most of the organism. RNA, though it also acts as genetic material in some viruses, it mostly functions as a messenger. It even has additional roles. That is, it functions as an adapter that is in tRNA, as an adapter molecule in tRNA, as well as tRNA. Then structure, and in some cases catalytic molecule. Which catalytic molecule you have learned in biomolecules? It is ribozyme. So DNA acts as a genetic material for majority of the organism. RNA acts as genetic materials for viruses, in some viruses, and it mostly functions as a messenger. Then RNA has other additional roles as well. That is, it functions as an adapter in tRNA, structural, and in some cases a catalytic molecule such as ribozyme. In class 11, you have already learned the structures of nucleotides, the way these monomer units are linked to form nucleic acid polymers. In this chapter, we are going to discuss the structure of DNA, its replication, process of making RNA from DNA, that is transcription, the genetic code that determines the sequence of amino acids in protein, and the process of protein synthesis, translation, and elementary basis of their regulation. Now the determination of complete nucleotide sequence of, so what are we going to learn in this chapter? We are going to learn the structure of DNA, how DNA, that is, replicates. Then the process of making DNA from RNA, making RNA from DNA. So while transcription, that is, your DNA dependent RNA polymerase makes RNA, which is complementary to the that is pairing of DNA, complementary base pairing, that is, it makes mRNA. Then genetic code that determines the sequence of amino acid, that is codons in proteins, the process of protein synthesis, translation, which happens in your tRNA adapter molecule, and elementary basis of their regulation. The determination of complete nucleotide sequence of human genome during last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will be discussed. Let us begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In subsequent sections, you will understand that why is it the most abundant genetic material and what is its relationship with RNA. So, the DNA. DNA is a long polymer of DNA is a long polymer of deoxyribonucleotides. DNA is a long polymer of deoxyribonucleotides. How will you define the length of DNA? The length of DNA can be either defined as the number of nucleotides or a pair of nucleotides referred to as base pairs. That is either you can say how many nucleotides are present or you can say how many base pairs are present, because one base pair will consist of two nucleotides. So the length of DNA can be defined as the number of nucleotides or the number of base pairs which are present in it. This is also this also is the characteristic of an organism. For example, so this is an important line. This also is the characteristic of an organism. So how? That is a bacteriophage, that is QX174, has 5,386 nucleotides, whereas bacteriophage lambda has 48,502 nucleotides, base pairs. E. coli or Escherichia coli has 4.6 into 10 to the power 6 base pairs, and a haploid human DNA has 3.3 into 10 to the power 9 base pairs. So how much does our diploid DNA have? That is 6.6 into 10 to the power 9 base pairs. That is 2n. This is n, 3.3 into 10 to the power 9 base pairs. E. coli 4.6 into 10 to the power 6 base pairs. QX174, 5,386 nucleotides, and bacteriophage lambda 48,502 base pairs. So into 2 if you do, then you will get the nucleotide. Similarly, if here 5386 you will do divided by 2, then you will get the base pairs. Now let us discuss the structure of such a long polymer. Structure of polynucleotide chain, that is, let us recapitulate the chemical structure of a polynucleotide chain, that is DNA or RNA. A nucleotide has 3 compounds. What are those 3 compounds? Firstly, nitrogenous base, then 5 that is pentose sugar, 5 carbon sugar, for example ribose in the case of RNA and deoxyribose in the case of DNA, and a phosphate group. There are 2 types of nitrogenous bases, purines and pyrimidines. Purines are adenine and guanine. You have to remember all this. Purines are adenine and guanine, and pyrimidine are cytosine, thymine, and uracil. Now, cytosine, adenine, and guanine, all 3 of them are common for both DNA and RNA, whereas thymine is only present in DNA and uracil is only present in RNA, and it is present in the place of thymine. Thymine is also called as 5-methyl uracil. And due to this presence of thymine instead of uracil in DNA, DNA is more stable than RNA. And what is the difference between that is your DNA and RNA, that is pentose sugar, that is deoxyribose and ribose, that is in DNA, in the second position of DNA, that is if we draw the pentose sugar, that is in the second position, there is only H, that is there is no OH, whereas in RNA here in this position there is OH. So because here oxygen has been removed, therefore we call this as deoxyribose sugar, whereas over here since OH is present instead of that is H in RNA, we call it ribose sugar. Now a nitrogenous base is linked to the OH of the first carbon pentose sugar through N-glycosidic linkage to form a nucleoside such as adenosine or deoxyadenosine. So a nitrogenous base is linked to the first carbon the pentose sugar through what? Through a N-glycosidic linkage to form a nucleoside. So to the first carbon, a nitrogenous base is linked to form a nucleoside. That is, what is nucleoside? It is only sugar plus nitrogenous base. When does it become a nucleotide? That is, and this bond between them, it is N-glycosidic bond. N-glycosidic bond. Between a sugar and nitrogenous base, the bond formed is glycosidic linkage. Glycosidic linkage, why? Because minus H2O, that is, water comes out and glycosidic bond is formed, and it is called as N-glycosidic linkage. And what do they form? Both of them together, that is without any phosphate group, that is nucleoside. Nucleoside is adenosine, deoxy or deoxyadenosine, then guanosine or deoxyguanosine, then cytidine or deoxycytidine, and uridine or deoxythymidine. Okay, so what are the names of nucleotides? This is very important. Adenosine or deoxyadenosine, then guanosine or deoxyguanosine, cytidine or deoxycytidine, uridine or deoxythymidine. When phosphate group is linked to the OH of 5 carbon of a nucleoside, so here when phosphate group is linked to the 5th carbon, then it forms a nucleotide, and this is by through a phosphoester bond, that is phosphoester linkage, and a nucleotide or deoxynucleotide, depending upon which sugar is there, it is formed. Two nucleotides are linked through 3,5 phosphodiester linkage. So again, phosphoester bond is also formed by losing of one hydrogen, but when you are linking two nucleotides with each other, then in that case, that is this, so this is the first carbon to which your guanosine is attached and nitrogenous base is attached. So this will become your N-glycosidic linkage. Then this is your 1, 3, 4, 5. Your 5th carbon to which your phosphate group is attached. So this is phosphoester bond. And this is your 3rd carbon to which another phosphate group is attached. This is phosphodiester bond. So when phosphate group is linked to OH of 5th carbon of a nucleoside through phosphoester bond or phosphoester linkage, then the nucleotide or deoxynucleotide is formed. When two nucleotides are linked through 3,5 phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. So therefore a polymer formed has one end free, so one will be having a free phosphate group, that is, if you see from this, that is 5' to 3'. Then in one part you will have a free phosphate group, and in the other part you will have a free that is the OH group. Phosphate moiety, that is free phosphate moiety at 5' end, which is referred to as 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH at the 3rd carbon, which is referred to as 3' end of the polynucleotide chain. Backbone of a polynucleotide chain is formed by sugar and phosphate. The backbone of a polynucleotide chain is formed by sugar and phosphate, whereas the nitrogenous bases linked to the sugar project from the backbone. So these are protruding. The nitrogenous bases, they project from the backbone. This 5', 3', 5', 3'. Here a phosphate group is free. Here OH group of the carbon ribose or deoxyribose, that is that is it is free. Then third carbon, then here again the third carbon, the OH is free, and here again your phosphate group is free. Then here there are nucleotide, the nitrogenous base, nitrogenous base, nitrogenous base, nitrogenous base, nitrogenous base, nitrogenous base, nitrogenous base. They are all forming, that is, nucleotides. So here there is your pentose sugar, and here is your phosphate group. This phosphate group is linked to the third carbon of this pentose sugar. So here it will be joined to the fifth carbon, and here it will be joined to the third carbon. In RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. Also in RNA, uracil is found at the place of thymine. It is called as thymine is also called as 5-methyl uracil, like I said. So in RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. In RNA, every nucleoside residue has an additional OH group which is present at the 2' position of the ribose. Whereas in DNA, there was no OH, there was H in the 2' position of the ribose. That is second position of the ribose. DNA is an acidic substance present in the nucleus. When was DNA first identified, and by whom, and what was it called? So DNA was first identified by Frederick Miescher in 1869, and he named it as nuclein. However, due to technical limitation in isolating such a long polymer, that is the elucidation of structure of DNA remained, that is it remained elusive for a very long period of time. It was in 1853 that James Watson and Francis Crick, based on the X-ray differentiation data which was produced by Maurice Wilkins and Rosalind Franklin, they proposed the very famous and simple double helical model or double helix model of DNA. It was one of the hallmarks of the proposition of the double helical structure by James Watson and Francis Crick was base pairing between the two strands of polynucleotide chain. However, this proposition was also based on the observation of Erwin Chargaff. For a double-stranded DNA, the ratios between adenine, thymine, guanine and cytosine are constant and they all equal 1. That is 1:1 ratio. Chargaff said 1:1 ratio. The base pairing confers is a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore if sequence of bases in one strand is known, then the sequence in the other strand can be also predicted. So if one strand I know it is A, T, G, C, then over here it will be T, here it will be A, here it will be C, here it will be G. So because the base pairing confers a very unique property of the polynucleotide chain, and they are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the other can be found out or predicted. If each strand from a DNA acts as a template for synthesis of a new strand, the two double-stranded DNA thus produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very, very clear. So the base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore if the sequence of bases in one strand is known, then the sequence of other strand can be predicted. And also if each strand from a DNA, let us call it as the parental DNA, acts as a template for the synthesis of a new strand, then the two double-stranded DNA thus produced would be identical to the parental molecule. Because of this, the genetic implications of the structure of DNA became very clear. So if each strand will act as a template, so let's say this was my first strand, this acted as a template for one molecule, this acted as a template for the second one. So I will get over here, I will get TACG, and over here I will get ATGC. So they both became now complementary, right? Because these both are complementary to each other. So इसका complementary और इसका complementary भी एक दूसरे से complementary होंगे. तो तो इसीलिए if so therefore the new two strands which would be formed, they would also be, that is, identical to the parent DNA. And because of this, the genetic implications of the structure of DNA became very clear. So what are the salient features of the double helical model, that is, which was proposed by that is Francis Crick and James Watson in 1953? They are as follows: that is, the DNA is made up of two polynucleotide chain where the backbone is consisted of sugar and phosphate and the bases are projected inside. Then the two chains have anti-parallel polarity, that is, they both are anti-parallel to each other. One is 5' 3' then the other is 3' 5', and then the bases in two strands are paired by hydrogen bonding, that is forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three hydrogen bonds. As a result, always purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. The two chains are coined in right-handed fashion. The pitch is 3.4 nanometer. A nanometer is 1 billionth of that of meter, that is 10 raised to power minus 9 meter. There are roughly 10 base pairs in each pitch. In each turn. Consequently, distance between a bond pair in a helix, that is 0.34 nanometer. Distance between the two strands is, I think, 2 nanometer of sorry, wait. Yeah, 2 nanometer. It is 2 nanometer. Distance between the two strands is 2 nanometers. Between two base pairs is 0.34 nanometer. One pitch is 3.4 nanometers. And there are 10 base pairs, that is, in each turn. The plane of one base pair stacks over the other in a double helix, and this, in addition to hydrogen bond, confers the stability of helical structure. That is, the plane of one base pair, it stacks over the other in double helix. Therefore, addition to the hydrogen bond, this also gives stability to the helical structure. Compare the structure of purines and pyrimidines. Can you find out why distance between two polynucleotide chain remains almost constant? This purines have purines are double ringed, and pyrimidines are single ringed, and also purines have double bond, whereas pyrimidines are single bond. And because of this, that is, this helical structure is maintained. The proposition of a double helix structure for DNA and its simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in molecular biology, which states that genetic information flows from DNA to RNA to protein. So first, in DNA itself, replication takes place, then RNA acts as a hacker, and transcription takes place, that is, mRNA is formed, and then from mRNA, that is, proteins are made by translation. This is the central dogma. And what did Francis Crick said? Francis Crick said that it is always going to be unidirectional in nature. This was his statement, that is, that central dogma of molecular biology, it is going to be unidirectional in nature. But there was one exception, that is viruses, that is RNA to DNA, that is reverse transcription took place. And what is the example? Reverse transcription. reverse transcription took place, and example was HIV/AIDS. Packaging of DNA helix. Distance between two consecutive base pairs, 0.34 nanometer, which is 0.34 into 10 to the power minus 9 meter. If the length of DNA double helix is typical mammalian cell is calculated by multiplying the number of base pairs with the distance between the two consecutive base pairs, that is length of the DNA that is in mammals that is diploid is 6.6 into 10 to the power 9 base pairs into how many base pairs are there, 0.34 into 10 to the power 9 base pair. Then what is the that is approximate and complete length of a DNA? It is 2.2 meters. This is very important, 2.2 meters, and a length that is far greater than the dimension of a typical nucleus, which is approximately 10 to the power minus 6 meter. Then how is it packaged in a cell? If the length of E. coli DNA is 1.36 millimeter, can you calculate number of base pairs in E. coli? That is, we know 4.6 into 10 to the power into 10 to the power 6 base pairs are present in E. coli. So 1 point, it's seeing E. coli DNA has 1.36. Can you calculate number of base pairs? So 0.34 into 10 to the power minus 9 into x is equal to 1.36 into 10 to the power minus 3. Minus 6. 136 by 34 into 10 to the power minus 6. 2, 6 are 12, 13, 8 are 16, 2 under 2, 3, 7 are 14, 7, 4 are 20, 8 are 6. 4 into 10 to the power 6 is the answer. How? That is, we know that 0.34 nanometer is the distance between two base pairs into the that is into the length of the that is number of nucleotides will give us the total length of the DNA, and here length of the DNA is given and base pairs is asked. Even though we read that E. coli has 4.6 into 10 to the power 6 base pairs, still here the length is different, so they are asking how many that is base pairs will be there. So we got it by this. In prokaryotes such as E. coli, though they do not have a defined nucleus, DNA is not scattered. How it is in DNA being negatively charged is held with some proteins which are positively charged in a region which is termed as nucleoid. DNA nucleoid is organized in large loops which is held by protein. In large loops which is held by protein. In eukaryotes, that is in eukaryotes, this organization is much more complex. There is a set of positively charged protein which are basic. These are called as basic proteins which are called as histone. A protein acquires charge depending upon the abundance of amino acid residues with charged side chain. Histones are rich in the basic amino acid residues lysine and arginine. So the basic histone proteins, they are, that is histone octamer. It is made up of your DNA, that is long polymer DNA, wrapped around the positively charged histone octamer. Why? Because histones are, why are they basic? Because they are rich in the basic amino acid residue lysine and arginine. Both the amino acid residues carry positive charges in their side chain. They are organized to form a unit of 8 molecules, which is called as histone octamer. Negatively charged DNA is wrapped around positively charged histone octamer to form nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. This is very important. That is, a typical nucleosome contains 200 base pairs of DNA helix. Then nucleosome constitute repeating unit of a structure in a nucleus called as chromatin, which is also called as beads on a string. Then seen in nucleus. The nucleosomes in chromatin are seen as beads on a string structure when viewed under the electron microscope. This is how they look, like beads in a string. The beads on a string structure in a chromatin is packed to form chromatin fibers that are further coiled and densed and metaphase to form cell division to form chromosome. The packaging of chromatin higher level requires a set of protein which are called as non-histone chromosomal proteins. Okay, next we'll do the second part. We'll revise this and then we'll do the second part in the next video. Thank you.